Hello, everybody, and welcome to the latest installment of the CDX Engage webinars hosted by Biodesix. This webinar is entitled Advancements in Proteomics and Machine Learning Techniques for Diagnostic Test Discovery. This is an online event designed to bring you with the opportunity to explore how new technologies and statistical methods are enabling us to further understand the biological underpinnings of diagnostic tests. To briefly introduce myself, I am Noemi, a member of the CDX Engage team here at Hanson Wade, and I will be chairing today's session. The webinar today will consist of one presentation, followed by a Q&A session where you will be able to ask your pressing questions to the speaker, together with some quick interactive poll questions to let you provide your feedback. Please remember to submit all of your questions via the Questions tab at the bottom of your webinar dashboard. And please remember you can submit this at any time throughout the webinar. We will try to answer all questions in the allotted time, but all unaddressed questions will be passed along to Biodesix so they can follow up with answers directly. Our expert speaker for today is Robert Georgantas, Senior Vice President of Research and Translational Science at Biodesix. Robert has served as Senior Vice President of Research and Translational Science in Biodesix since August 2019. From 2014 to 2019, prior to joining Biodesix, Dr. Georgantas worked at AbbVie, where he served as Director of Immunology Programs and Biomarkers within the Genomics Research Center of Excellence, a group of experts tasked with applying genetics, genomics, epigenetics, and metagenomics to inform the product pipeline primarily regarding new target discovery, biomarkers for clinical trials, and asset positioning. Dr. Georgantas completed his Ph.D. in pharmacology and molecular medicine at the Johns Hopkins University School of Medicine. But before moving on to Robert's presentation, I would like to first start with a poll to get everyone involved. You will have around 30 seconds to submit your answers. You will have around 15 more seconds. Perfect. Now moving on to the second question of our poll. What stage of development and pipeline are you focused on? Is it research? Is it preclinical, phase 1, phase 2, or phase 3? You'll have around 20 more seconds to vote. Perfect. Thank you everyone for your feedback in the polls just now. I don't want to keep you all waiting anymore, so now without further ado, I'd love to welcome Robert on stage to give his presentation. Thank you, Robert, for joining me.
Yeah, thank you. Good morning.
No
good morning. I'm Robert Georgantas. I'm Senior Vice President of Research and Translational Science here at Biodesix. Thank you for joining me today for this webinar discussing advances in machine learning explainability, as well as artificial intelligence transparency, and how we use these techniques to inform diagnostic test use, and potentially to dig into the background biology of a test.
Robert, one question, sorry to interrupt. We cannot see your screen. Can you let me know if not?
I will, there we go. Can you see now?
Perfect, we can see it now. So over to you, I'll leave the stage now.
Let's see here. All right, what is going on? So we'll start today by touching on the definitions of some different levels of artificial intelligence. The first level of artificial intelligence is our so-called expert systems and simpler AIs that mimic human behavior. Such things as industrial robots, anticipatory smart assistants such as autocorrect, and phase autocomplete when you're typing on your iPhone. At the next level is machine learning, a more complex subset of AI. These are so-called improving algorithms that improve as they iterate and incorporate more data. Machine learning includes things like spam filters in your email, low-level driving assistance, things that like lane departure assist, and some diagnostic applications. But even more complex are deep learning algorithms. These are multi-layer neural networks to employ complex pattern matching, to find a pattern that matches a disease. Examples include, advanced self-driving or full self-driving, image colorization, audio and video synthesis de novo, as well as advanced diagnostic tests. Biodesix Diagnostic Cortex AI platform falls into this deep learning category. There are other more complex forms of AI, such as adversarial networks, but we'll not discuss those today. Now, there are a number of pros and cons to AI. On the pros side, we have, they provide a very fast path to a solution because, they're basically you can run, thousands to millions of experiments simultaneously in an in silico environment. AI and machine learning platforms are able to discern patterns from very complex data that humans and traditional statistics cannot. They can handle semi-structured or completely fully unstructured data, meaning that you can put an outcome with your data to train to that outcome, or the AI can actually go in and use completely unstructured data and pull patterns out of that data. AI platforms can handle very large data sets, both in sample number as well as in feature number. Things at the order of anywhere from thousands to millions of subjects, looking at anywhere from thousands to tens to hundreds of thousands of different features. On the level of things like proteomics and genomics and transcriptomics. And they also have the potential to reduce human error. But on the con side, there are a number of potential problems. One is potential poor performance with small data sets, so-called short-wide data. This is something that we've answered with the diagnostic cortex using a technique called boosting. They're also very prone to overfitting, which is where you have a mathematic construct that perfectly fits your training data but doesn't generalize to other data sets and doesn't validate. We've answered this by using machine learning techniques like bagging, tuned specifically to clinical data sets so that our algorithm doesn't overfit. They're also prone to using meaningless differentiation. And by that, an example would be something like a study that was done, a machine learning algorithm that was developed at Mount Sinai a number of years ago that was looking at, when patients were brought into the ER, there was a machine learning algorithm that was trained to see if someone would progress, if their disease would progress to a life-threatening condition. And it turned out that the machine learning algorithm was only using whether or not someone got a portable chest X-ray. It wasn't actually using any other data to discriminate. So it wasn't actually giving a meaningful differentiation based on the available data. We answered this question by using extensive data curation and cleaning of the clinical data and omics data before it goes into our model. It's also prone to data biases, so-called edge cases, differentiation based on gender bias, racial bias, or socioeconomic overrepresentation. One way that we answer this Biodesix is by using training data sets that are representative of real-world data, and the actual patient population. But what we've been working on for the last couple of years has been the problem of transparency, or the black box problem of AI. Basically, this gives a new level of confidence in the outcome of AI, since we can now directly determine how the AI makes a decision, and is allowing us to dig into the biologic underpinnings of a test to assure that they make sense biologically. At Biodesix, we use a number of different technologies, including digital droplet PCR, next generation sequencing, standard mass spec-based proteomics, and our proprietary deep multi-proteomics to generate high quality data from liquid biopsies to profile a patient's tumor, their immune system, to do a general host profile, and we also incorporate clinical data. And all of that data goes through one of two pathways, either traditional statistics and bioinformatics to get to a diagnostic solution, because you don't always need AI to come up with a diagnostic test. But more commonly, that data, that high dimensional data is fed to our diagnostic cortex AI platform to develop a test which becomes a diagnostic solution. We've also built a number of modules into our diagnostic cortex, such things as protein set enrichment analysis, and we're exploring techniques including the seer proteograph and the new Bruker-Timstorff technology to identify the underlying biologic mechanisms that our test shows us. Now, one of the big problems with AI is the so-called black box problem. This is where you input data into the algorithm and you get an answer out, but you don't actually know what the internal mechanism is of the AI platform and what it's doing. Because these algorithms use complex pattern matching and multiple layers of data abstraction and representation, it's been thought that the internal behavior of the code couldn't be known. While there have been a number of successes with AI, this lack of explainability has contributed to many well-known failures of AI. I mean, if you just go to Google and type in failures of AI, you know, there are a very large list of things that come up where, you know, an algorithm was trained for an outcome, but it never actually worked in the real world. And we think that explainability will help answer this problem. Now, our AI research team at Biodesix has spent the last couple of years working to open the black box and achieve truly explainable deep learning algorithms by, you know, basically taking computational algorithms that go in and interrogate this black box and figure out what the AI is doing. Now, before getting into the specifics, we need to define two aspects of what we've been trying to achieve with our diagnostic cortex platform as far as explainability goes. The first being explainability itself, which refers to understanding the decisions made by the AI algorithm. Today, we're talking about proteomic-based diagnostics. This means which proteins and the patterns of those proteins are being used by the AI to make a diagnostic call. There are actually two different types of explainability. One is model explainability. It's focused on how the AI model interprets features, computational values, etc. Basically, the mechanics of how the model itself works. The other is predictive explainability. This is focused on why the AI model chooses an outcome. For example, our primary immune response test, one of our pipeline tests, how that would classify a patient as sensitive or resistant to a checkpoint inhibitor drug. Our primary concern is to use predictive explainability to give physicians and patients a better understanding of why our tests output their predictions. But our new explainability model that I'll go into in a moment achieves both model and predictive explainability. Now also, I'd like to talk about what we mean by transparent AI. Explainability is part of transparent AI, but there are a number of other facets of a diagnostic test that fall under the transparency umbrella and are actually provide clarity into test development and experimental design. These include transparency of training data sets to assure that they represent the real world disease population. In other words, when you develop a test, you want to assure that your training set is actually representative of the patient population, not some subset, not edge cases, not, you know, extreme responders, extreme non-responders. You want one that really models what we see in the real world. Another is visibility into data selection by the AI platform to assure that it's using data readily available in the real world. For example, you know, you don't want to use data that's not, it's, for example, if you're, you know, doing something with, I'll show a little bit later, a classifier that we came up with COVID-19 outcomes that uses real world clinical data, you know, from ER admissions to predict whether or not someone will go on to a severe outcome. In that case, what you want to use is you don't want to use data that's not available in the ER. You have to use data that's readily available and collected across the country in an ER for that model. If you have data in there that's not readily collected, your AI is not going to work. Another thing that you have to understand is any biases in your training sets, such as the weights of feature use in the algorithm, the so-called bias variance trade-off used to tune to overfit or underfit the model, as well as bias introduced by humans' preconceived notions. And then you also have to understand for transparent AI is visibility into model versioning to assure to stakeholders that the AI algorithm matches the transparency data that they are presented with for that model. And then finally, for diagnostic tests, an understanding of the biologic differences between prediction groups to assure that the predictions that are made by the algorithm also make biologic sense. So what we've done is we've taken advantage of a computational construct that was developed actually back in the 1950s by Lloyd Shapley. These are so-called Shapley values. So Shapley values analysis it's a subset of game theory that mathematically figures out the contribution of each player to an outcome. In the economic model that Lloyd Shapley used, the construct was used to divide winnings among a group of collaborating players with different abilities, who therefore made different contributions to the task at hand. The mechanistics of an AI algorithm are theoretically similar to this situation, in that the features or data fed into the AI algorithm will contribute differently to the prediction made by the AI. Therefore, we should be able to use Shapley values to explain an AI algorithm. Since Shapley values calculate the contribution of each data feature, or in our case, each protein, in the context of all other features, they give us the explainability that we desire. But that means that the computational complexity, but that means that the computational complexity increases exponentially as the number of features or proteins in your model increases. So computing them would become problematic. So we've actually implemented two different versions of Shapley value calculations in the diagnostic cortex. One that works out Shapley values exactly for classifiers with small feature numbers, such as our VeriStrat and our ARDS classifier, our COVID's outcome classifier. And another for classifier with large numbers of features that uses a polynomial structure to sample over feature subsets and thereby greatly reduce the computational time needed to reach what we call convergence or an actual definitive answer. Now, we recently published a paper describing the exact Shapley value method used with classifiers with small numbers of features. In this case, our VeriStrat classifier. In this paper, we demonstrate two very important aspects of the Biodesix Shapley value method. The first is that this is the only Shapley value method to achieve convergence, meaning that after a finite number of computational iterations, we get an exact repeatable answer for feature contribution. This isn't actually seen with other Shapley value methods that are currently used in machine learning. Second, the Biodesix Shapley value method is the only Shapley value method that meets all three Shapley value axioms: the axioms of symmetry, the null player property, as well as the axiom for additivity. And we won't go into any details of those, but please feel free, if you'd like more details, to contact us and we can go into what that means in greater depth. Now, the ability to explain AI becomes progressively more difficult over time, especially when we consider improvements in technology that allow deeper interrogation of a given omic. You know, from the genetic standpoint, there are over 5 million SNPs and hundreds of thousands of indels in the human genome that can be interrogated. For methylation, there are over 29 million potential methylation sites in the human genome. In RNA-seq experiments, it's common to observe over 65,000 unique elements or features of transcripts in an experiment. And now with our proteomics, we're approaching the data density of these other omics. For example, over time, our deep MALDI technique, in its first generation, we could look at, about 100 proteins replicably. In our second generation, which was used to develop some tests that we're using now that are in our pipeline, we could look at 274 proteins in their translational modifications in a replicable fashion. And now we've just entered our third generation of deep MALDI proteomics, where we can look at over 2,200 proteins and their post-translational modifications. And this is the platform that we're actually moving our pipeline test to, risk of recurrence, and our primary immune response test. And then we're also working, we're taking advantage of some new technologies, namely the Broker-Timstorff technology, as well as the seer proteograph technology, to develop what we call so-called, deep next generation platform, which, depending on the matrix, be it serum or plasma, where we can look at close to 3,000 proteins, as well as their isoforms, or to something like tissue, where we're achieving, where we can achieve greater than 9,000 proteins per sample. So you can see as time goes on and technology gets better and we get deeper into the genome or transcriptome or proteome, the problem of explainability becomes more and more complex. And I'll quickly go over 3 examples of different ways that we've used our explainability model to dig into our classifiers. The first being explainability around our VeriStrat test. Now, this is a test that identifies patients with poor and good outcomes, whether they're treated with chemotherapy, whether they're treated with immunotherapy. It's more of a prognostic test that looks at good and poor outcomes. And here we can see, looking at, this is an example looking at frontline immunotherapy versus chemotherapy. With no VeriStrat classification, basically the Kaplan-Meier curves look very much the same. There's no differentiation between the two. And if we look at VeriStrat classification, looking at just monotherapy with a checkpoint inhibitor, we see that the VeriStrat good outcome much outperforms those patients that are classified as VeriStrat poor with a hazard ratio of 0.45. And then if we look at those patients treated with frontline immunotherapy plus chemotherapy, we see, with VeriStrat classification, we see a very large differentiation of the patient population. Showing those with a VeriStrat good, they don't meet a 50% median survival. They do much better than the VeriStrat poor patients with a hazard ratio of 0.23. Now, if we go and we want to look at, what feature use looks like within a VeriStrat population, what we see is the way that we've chosen to represent this is by using radar plots, where the center is 0 contribution of the feature. The edge is, maximal contribution of the feature. So it's, between, on a scale of 0 to 1. With blue representing that particular feature going towards a good outcome, and red representing that feature going towards a poor outcome. And what we see here is that we see different patterns of feature use depending on the patient. So we have so-called uniform goods, where all of the different features of VeriStrat are contributing to the outcome towards the good, in the good direction. With about equal contribution. On the opposite side, we have the uniform poors, where we see the opposite of that outcome, where all of the features are contributing pretty much equally, but in the poor direction. But then we have cases in the middle, where we have what we call the non-uniform good and the non-uniform poor, where these patients show diverse feature use. And these are just a couple of examples. Here, this patient is showing some features that trend towards the good outcome, some that trend towards the poor outcome. But when we add these up, when we add up the contribution of these features, we see that 2 features, this 11-15-29 and 11-6-85, are contributing quite a bit to the outcome. But when we add these up, we see that the additive score of all of these leads to a good outcome call. Whereas on the other side, we see this other patient that's in what we call a non-uniform poor, where those 2 major features are trending in the opposite direction towards a poor outcome. But we do have other features that trend towards the good outcome, but with less contribution. When we add these up, what we see is that this patient is called a poor based on feature contribution. Another example is that of a set of classifiers that we came up with around COVID-19 outcomes. Basically, this classifier was developed. This is also a published classifier in this reference, the reference shown here, where what we were able to do is we were able to take clinical data. We started with a little over 300 clinical features from the emergency room, from when patients were taken into the emergency room with COVID. And what this classifier was developed to do was to assess risk of whether the patient would go on to develop a severe outcome, whether that was acute respiratory distress syndrome, whether that was being intubated, or being admitted to the ICU. What we were able to do here is we were able to come up with this classifier that put patients in a low risk category, a lowest risk category, a low risk category with a 99% and a 92% probability, and then patients that were in intermediate risk or higher risk. And basically what this classifier did is it identified those patients at the lowest risk so that they could be sent home, they could be monitored less. It showed that they weren't going to have a poor outcome. And when we look at feature use of this, some interesting things came out of looking at feature use for this particular classifier. One is that we started with over 300 features fed into the diagnostic cortex. We initially feature reduced that down to 57 features. And one of the things that the Shapley values allowed us to do is what we call cogent feature selections. Not only does it show us the different feature usage by the different classifiers, so the initial split where we go and we split patients into low risk and high risk, and here these graphs are showing the feature usage of all some of the different features that went in, and then going to and then splitting those again into the lowest risk and low risk and high risk and highest risk. The other thing that allowed us to do is it allowed us, by identifying those features that were contributing most to the algorithm, it allowed us to further feature reduce down to 26 features that were used in the algorithm overall, between clinical lab results as well as demographic information and emergency room numbers. So, this was one application where we can use Shapley values by looking at the actual contribution of the features to go and do more cogent feature selection rather than what's done nowadays. What's done normally is just random feature selection where you iterate over and over with different feature combinations. And then if we dive down into the actual feature usage for an outcome of different patients, at the individual patient level, what we can see here is, again, looking at those radar plots, what we see is really significant differences in feature usage between a patient that's the lowest risk or a patient that's in the highest risk category by looking at these different radar plots. So for the first split, here we have what we call the tree for splitting patients out, and then the actual overall representation of what the feature use looks like for a patient in the lowest group versus a patient in the highest group. And what we see here is that a patient in this particular patient was chosen to be in the low group due to a combination of their age, their CRP level, and the BUN level, as well as CRP and ferritin at the next split of differentiation. Where this patient in the highest risk level, in the highest risk category, was classified based on their age, their CRP level, their BUN, their race and gender, as well as their LDH level and oxygen saturation. So what this demonstrates that at a per patient level, the algorithm's actually using different features to make a call depending on the outcome. And then finally, our final example here is really our primary immune response classifier that identifies 3 different response groups to patients treated with checkpoint inhibitors. So those that fall into a highly resistant group, those that fall into an intermediate group that are more likely to be responsive to either a combination therapy of a checkpoint inhibitor with with platinum drugs, or a patient that is responsive to monotherapy. And here we see great differentiation for OS between the good group and the bad group, with that outcome a hazard ratio of 0.15, showing a really strong differentiation for OS as well as PFS. We don't see the patients in the good group reaching 50% overall survival. So they never meet the 50% median. And when we look at feature usage in these patients, what we see using our Shapley value technique is we see that not only do we see the contribution of the different features here for those patients making with a pure resistant call versus a pure sensitive call, we see a couple of different things. One, we see, if we look at the actual features that are contributing to these calls, there are some similarities and there are some differences. The other thing that we see here, we're just looking at the top 22 features for each call. We see that there are a number of features out of the 274 that were used to develop this test that are making a significant contribution to either the resistant call or the sensitive call. But the other thing that was really exciting that came out of this data set is that it allows us to start digging into the biology of what this test is doing. And we see that here in the red box. This is a feature that we've been able to identify. It's feature number 10848. This actually turns out to be an isoform of the S100A8 protein. Now this is a known biomarker along with its partner S100A9. Those proteins partner together to become a biomarker for basically immune activity. This is a really well-known biomarker in the autoimmune space. And this particular isoform of S100A8 had the largest contribution between the sensitive group and the resistant group here to making a call for whether or not the patient was resistant to a checkpoint inhibitor. So not only can we look at the feature use, we can identify which features are most important for the call. We've been able to show with this particular experiment that multiple features contribute to that call, but then we're also able to start digging down into the biology of what these features really mean and how they play into the classifier. And then there are a couple of other implications of Shapley values that are really, really interesting. Number one is, one of them is the idea that we can actually use Shapley values and identifying these subsets of feature use to actually go and dig into each of those classifier arms and use Shapley values as a potential subclassifier. And here what we're showing is a data from a drug that we were developing a classifier with a partner for a specific drug, where basically what we did was similar to the primary immune response classifier. We had patients that were in a good, that were sensitive to the drug versus resistant to the drug. And here what we were able to do is we were actually able to take those patients that were poor response, run Shapley values for each individual subject in the study, and then look at the Shapley value, feature use, for that outcome. And what we identified is we identified two groups within that poor response group to the drug. Here, response group A, which show one pattern of feature use, and then response group B that shows a different pattern of feature use. And the interesting thing is that when we take that data, those two different response groups, and look at their outcomes, we're actually able to then go and split that poor response group into one group that responds a little bit better to the drug and one that's very close to completely resistant to the drug. Now, we're not quite sure what the implications of this are yet. One of the things that we would love to be able to do with data like this is take that and tie it to, for this particular drug, does this group then get drug versus not get drug, to see, because we do have some responders here. The other thing that we're doing is using this type of analysis with our internal classifiers to see if we can link these specific outcomes for different classifiers to a specific drug. So, will this aid in, prognostic use? Will this aid in therapeutic guidance as to which drug to use for an outcome? The other implication of this is that, we feel that, what we're actually seeing here, because we're looking at a large proteomic panel of over 274 proteins, is that we may actually see that this difference may actually represent different biologies that lead to resistance to the drug. So this may be one biologic mechanism versus another biologic mechanism that both have the same phenotypic outcome of being of drug resistance. So the other implication is that we should be able to use Shapley values to do a more cogent look into analysis of disease biology. And by that, what I mean is taking a patient cohort, using our diagnostic cortex or any AI platform really to, go and divide that group into different outcomes, then use Shapley values, because, we're using, with our previous generation of deep MALDI, our deep proteomics, we were only looking at 274 proteins. But now we're, looking at over 2,000 proteins in their post-translational modifications, letting us dig even deeper into the biology. We can use then our exact Shapley value, our Biodesix Shapley value analysis, to then go and subset those groups into what we think are probably different biologies. So this example here, we'd be looking at, one biology here, another biology in this subset, and then potentially a third biology in this other pattern that we see in the heat map. Then take those subjects and subset them down. So let's say that these are all from a resistant group. We then use the seer proteograph technology that we have in-house, as well as the Bruker-Timstorff, to identify differential protein expression amongst these groups. That then allows us to go and do standard data analysis, where we can do, look at differential protein expression, or if we're using another omic like genetics or transcriptomics or epigenetic data, break those down into different analysis groups, and then do standard bioinformatics, things such as pathway analysis and upstream driver analysis, to identify the biology behind why we're seeing these different resistance mechanisms, as an example. And with that, I'll end with this slide and thank everyone for attending the webinar, and then take questions.
Hi, Robert. Thank you so much. If you give me 1 second, there you go. I just want to remind to the audience that if you have any questions for Robert around what he just presented, please remember to submit this using the questions tab at the bottom of the webinar dashboard. But before moving on to the Q&A session, I have 2 more poll questions. So same as before, you'll have around 30 seconds to submit your answers. So are you interested in learning more about the application of Shapley values to diagnostic tests? Yes or no? You have around 10 to 15 more seconds. Perfect, thank you everyone. And finally, the last poll questions for today. Do you have a program that could benefit from this type of approach? Yes or no? I'll give around 10 to 15 more seconds. Perfect. Well, thank you so much, everyone, for voting. It's lovely seeing so much interaction and participation from the audience. So now, as mentioned before, we will be moving on to a Q&A session where you'll be able to ask any questions you might have for Robert after the presentation he just gave. So, Robert, thank you for your presentation before. It's a pleasure having you here today for this Q&A session. I see we have some questions coming through. So the first question is, are the features being described a mix of EMS identified proteins, methylation events, and PTMS?
So for this presentation, what we were looking at was proteomic data, and it was a mix of everything that I showed in this slide except for the COVID-19 outcome classifier, was proteomic data from our Deep MALDI platform. So it's whole protein proteomics, looking at the protein expression as well as post-translational modifications of those proteins. And as I talked about, some of the older classifiers that we were looking at, there were about 274 proteins in their PTMs in that data. With the current generation that we're using, such as for the primary immune response classifier that I talked about, we're looking at we can assay up to 2,200 proteins in their their PTMs as well. The clinical outcomes classifier, the COVID-19 clinical outcomes classifier, was based solely on data collected in emergency rooms. So as intake data when patients were presenting with COVID-19 at the emergency room. But we are able to do multi-omics examinations with this data, where we can, for example, our risk of recurrence classifier incorporates a number of different clinical features along with proteomics data. So we're able to do this at a multi-omic level as well.
Perfect, thank you. And we have another question that says, what were the specimen types here? Or what was the specimen type here? So I'm guessing
the specimen type was they were serum samples. So they were retrospective serum samples that we were looking at from bank samples. In practice, we have a blood collection device that we use for serum collection in the clinic. So.
Perfect, thank you. And another question just came through. How does assay reproducibility vary based on the assumed pre-analytic steps involved? For example, extraction or trypsin digestions?
Yeah, so we see very little variability with the pre-analytics. We're, especially with the Deep MALDI technique, we do a great deal of of optimization of the assay for collection of the sample, as well as processing the sample to bring down variability as much as possible. Now on the bottoms up side, with trypsinization, we see the standard variability that you would see with trypsinization to do peptide-based LC-MS. But again, that's as controlled as much as possible.
Perfect, thank you so much, Robert. And what are the next steps in your process after Shapley Values, and why does this matter?
Oh, so the next steps after Shapley Values, I mean, obviously we've been doing this research into explainability and transparent AI for about 2 years now. And while we know that we've come up with an extremely useful tool in Shapley Values, especially since we've got it to the point of convergence, meaning that when we run the same analysis over and over, we get the same answer, unlike other methods of explainability that are out there. We're currently exploring a number of different avenues to explainability. Shapley values aren't the only mathematic construct that are out there. The other thing that we're very actively exploring is the concept of causal inference around the outcomes of our classifiers. So the difference there is Shapley values and the explainability algorithms show us what features are being used and how they're being used within the algorithm. What we'd like to be able, the next step that we'd really like to take is to use the concept of causal inference to go in and say, start to computationally look at, if protein X is differentially expressed within our experiment, is that actually a protein that is contributing to a disease versus a protein that's just a biomarker of a disease? That's really where we'd like to go next, and and start developing causal data around the informatics that we do, rather than just explainability data.
And now that we're talking about Shapley values, like how can this help in the communication to physicians?
Well, I think that there's a lot of value in explainability in the AI. I've talked to a number of physicians that are simply not big believers in machine learning and AI because they don't understand how it works, and no one can explain to them how that test works. And I think that it may not make a big difference in the outcome of how a test is used, but I think that it gives physicians and patients and payers and other stakeholders much more confidence that that your test is actually working. Because if you can explain how it's working, if you can explain the biology behind the different outcome groups, it gives a lot more confidence to those stakeholders that the test is actually showing them something meaningful. So it's more of a tool to help a physician understand why a test is showing them what it's actually telling them a risk profile is. And I think that's good along the board, especially with the number of the failures in AI in medicine that have recently been highlighted around the COVID-19 pandemic. In all honesty, some in the oncology space, some in the autoimmune space that just have worked really well in training sets but haven't gone to the real world. So that explainability just helps us explain what the algorithm is doing and gives us surety that the algorithm is showing us something that's biologically meaningful.
And I see another question coming through. Are these tests offered as a centralized service or as decentralized IVD?
These are all the tests that I showed today are LDTs, so they're lab determined tests. They're not IVD tests at this point.
Perfect, thank you. And going back to your presentation, what do you mean exactly by transparent AI?
So transparent AI is more the concept. So explainability is the concept of knowing basically what the gears are, and the math inside your algorithm, what's going on inside the algorithm. Transparent AI is a much broader concept around not just understanding what the algorithm is doing, but understanding all of the data that went into the algorithm to build that algorithm. So transparent AI includes things like understanding what the actual training set was to assure that you actually have a real world population that you're training the data to. It has things in there like understanding what the biology is behind the test out, the outcome groups, to show that your algorithm is biologically meaningful. It's a much broader concept in understanding the entire process, so that you have transparency into seeing what the different biases are, and the trade-offs are that went into making the algorithm, to give even more surety that the algorithm is going to be useful for your patient, or your disease of interest.
Perfect, thank you, Robert. And I guess my last question would be, how are Shapley values used to perform biomarker ID if the features in MALDI are still undefined?
Oh, so the nice thing is that another nice thing about Shapley values that I didn't really touch on in this talk is that, with MALDI, we only know, like for the S100A8 protein that I showed earlier was one feature in the MALDI spectra that we know what the protein is. For many of the proteins, we haven't identified, or for many of those features in the spectra, we haven't actually identified what the proteins are. So one of the ways that Shapley values really aids us is, without knowing what the protein is, it allows us to say which of those features are contributing to the diagnostic outcome. And it allows us to then go and focus on, instead of, especially with our newest version of Deep MALDI, where we're looking at over 2,200 proteins, for any given classifier, any given test, it allows us to go in and really focus on just those features that are being used by the AI to make a decision. When we then go and use something like the Bruker-Timstorff and the SEER technology to go and identify what that actual spectral feature, what protein is being represented by that. So it lets us hone down, rather than looking at the entire spectra, just looking at those subset of proteins that are making the decision, for further biologic characterization.
Perfect. Thank you so much, Robert. I don't see any more questions coming through, so I think we're going to wrap up now. But any final thoughts, Robert, key takeaway messages for the audience you want to give before the end of the webinar? Like, if you had to summarize your presentation with 3 key takeaway home messages, what would this be?
I think the key message would be that, we've, for our diagnostic cortex platform, and, we're actually currently trying to extend that to other, machine learning platforms as well, and other deep learning platforms. With our Shapley value analysis, we've achieved a level of explainability that, others have not been able to do. And, we're really looking at this to, broaden the understanding of how our tests work, and, bring much more transparency to, how our diagnostic tests work.
Well, thank you so much, Robert, for the Q&A session and your presentation. Actually, I've just seen another question come through. So before wrapping up, what do you think about the resolution of RNA sequencing versus proteomics data?
Well, so there are 2 different things. So, the RNA-seq data obviously has, better resolution than current proteomic techniques, right? So you can look at, when I've run RNA experiments, we've come up with, hold on. Sorry, I'm daddy daycare today. I have a sick 3-year-old at home today who's asking me questions on the side here.
No worries.
When I've done RNA-seq experiments in the past, you can look at anywhere from 60,000 to 100,000 unique transcripts in an RNA experiment. So obviously, the resolution there is much better than what we see with proteomics, where we're seeing a maximum of about 9,000 proteins per sample. The disadvantages of an RNA-seq is that there seems to be much more variability in RNA-seq experiments than there are in the proteomic experiments, at least in our hands. And the other is that when you do an RNA-seq experiment, you have a lot of other things going on, like microRNA expression, long non-coding RNA expression, modifications to the RNAs that change what they do at the protein level. The outcome of that, at the actual phenotypic level. So, I think RNA-seq experiments and RNA expression is a great technique, but there are advantages of proteomics that you're actually seeing the end state, the actual phenotypic state, compared to RNA-seq. In all honesty, one of the things that we're working on currently is incorporating multi-omics into our classifiers, where we're looking at things like genetics, epigenomics, RNA transcriptomic data, as well as proteomics, to build better classifiers. So, I think that there are advantages and disadvantages to every omic. We can take advantage of each one to build better classifiers and get even more differentiation for patients.
So actually, the person that asked the question has replied. They've said, I agree. How complex is the quantification of protein expression using the platform you detailed?
How complex is the quantification? So in the deep MALDI technique, the quantitation of protein is relatively straightforward. You know, basically what we do is we look at the spectra and do an area under the curve integration to look at the level of protein that corresponds to the, you know, the different spectral features. The advantage of the technique that we're doing, you know, is, you know, our founder, you know, Heinrich Roeder, you know, he came up with a number of computational algorithms that allow us to take those spectra, which actually show, you know, quite a bit of variance in everything from peak width to baseline, you know, to, you know, the baseline of the spectra. He came up with a number of, you know, computational algorithms that, you know, perform, you know, spectral alignment, spectral normalization, baseline normalization, that make this you know, an extraordinarily you know, consistent measurement. You know, we see, you know, for most of the proteins that were, or features, I'll say features that we're measuring, you know, we see you know, CVs of less than 10%, with the vast majority of the features being, you know, having CVs of less than 5%. Now on the other side, you know, when we look at you know, the bottom-up proteomics, when we're doing, you know, an unbiased look, you know, but at the peptide level, you know, we do see more variability than that. You know, we see, but you know, what we're currently doing with the bottoms-up proteomics is we're attempting to take those computational algorithms that are used in deep MALDI and apply those to the spectra that we get from bottoms-up proteomic to normalize those as well, you know, and get those, you know, really maximize the CVs for that technique as well. But that's a work in progress.
Robert, can you hear me? I'm so sorry. My laptop just froze.
Uh-oh. I can still hear you.
Okay, perfect. I'm not sure if you can see me because, as I said, my laptop just froze.
No,
can you remind me of the last question you just answered?
The person was asking about variability of the platform and quantitation of protein, you know, actual quantitation of protein measurement.
Okay, perfect. Thank you so much, and apologies. So we do have another question: is do you need to tweak the algorithm as you add more proteins into the system?
We do not. No, we don't. The AI, you know, the diagnostic cortex, as well as the you know, the Shapley value module of that are insensitive to, you know, the number of features that we put in. What does go up is, obviously, as you know, you add more features, computational time goes up. You know, so an active area of research that we have going on in our bio, you know, in our AI team, are ways to speed up, you know, the algorithm. You know, either by, you know, migrating it to different platforms, you know, looking at ways to do much more parallelization, as well as, you know, moving from, you know, currently we're on a CPU structure and we're moving to a GPU structure, you know, to speed up the computational time.
Perfect. Well, thank you so much, Robert, and apologies again because I don't know what happened to my laptop. But we are running out of time, so I just want to thank the audience for submitting those great questions. And Robert, thank you for your presentation and for joining me on the Q&A. Please feel free now to leave the stage.
Great. Thank you, everyone.
As I said, this is all the time we have, sadly, but on behalf of CDX Engage and Biodesix, I really hope you enjoyed today's webinar. Please look out for an email tomorrow from us, as we'd love to hear from your feedback on today's webinar. And if you do have any follow-up questions or comments, please email this to myself at info@hansenwade.com, and I will get back to you soon. As I said, on behalf of Biodesix and CDX Engage, thank you all for your time, and I hope you enjoy the rest of your day. I really hope to see you next time.